Thank you for that really nice introduction, Professor Feldberg. And thank you to the foundation and the organizers for inviting me to present today. So I think it was great that we had a lot of really nice IVF talks already and assisted reproductive talks, because I'm probably going to go more from the epigenome perspective. So every cell in our body has the one genome, but we have hundreds of epigenomes. And one of the main reasons for this is that we have 2 meters of DNA that needs to fit into a cell nucleus that is 100,000 of a meter in diameter. So to do this, DNA is compacted. And at each sort of layer of this compaction, we can have modifications. So at the very sort of beginning, we can have DNA methylation at the level of DNA. We can also have non-coding RNAs. And then at the unit of the chromatin, the nucleosome, we can have histone modifications. And all the way to more tertiary structures, the most complex of which is the mitotic chromosome. So from this one genome, we can have many different cell types, all with the same DNA sequence, but a different phenotype and a unique epigenome. So our understanding of the genome started in the 90s and culminated this publication in 2001 from the Human Genome Consortium, and we had the genome sequence. But it's only 20 years later that we actually completed the whole genome, and I think that was touched upon earlier today. And sort of currently what's being sort of driven is to make the genome more inclusive. So what research efforts are doing now is trying to sequence as many diverse genomes as possible. But even so, even in 2010, it was evident that even if we sequence everybody, we still won't know everything, because there's a whole other layer of information above DNA that we call epigenetics. So the efforts to understand the epigenome also started sort of very quickly after the genome was published. And these sort of fall under this giant International Human Epigenome Consortium, which is sort of just an umbrella term. And under this sort of consortium, there are many different projects all around the world. It is a big consortia that include a lot of different labs in different countries, including ENCODE, probably the most popular, famous, and currently the Human Cell Atlas. And I spent 4 years working on the Blueprint Epigenome Consortium. And the aim of this consortium was to publish reference epigenomes, that's what we call them. And the reference epigenome was to have 5 histone marks out of the total 200 plus, whole genome bisulfite sequencing, transcriptional, like RNA-seq, and open chromatin. And the idea was that this reference epigenome would tell us something about cell identity and would tell us what, or at least would be able to predict cell function. So in our lab, some of the techniques we use to analyze the genome is RNA sequencing to look at the gene expression, RTAC-seq to look at where the nucleosomes are. We use cut and tag to look at histone marks. So the idea there is to see which parts of the genome are active or inactive. Whole genome bisulfite sequencing, or EPIC arrays, which I'll talk about today, to look at DNA methylation. And there's another whole layer of information using chromosome capture confirmation, which tells us which pieces of DNA interact with each other. And what's really nice is sort of over the last 10 years, we went from needing a lot of material to do these assays, and now you can do most of them at single cell level. So what can this sort of data tell us? So the whole sort of aim of the lab is to integrate this epigenomic data together to see if we can explain biology. So if you think of this as a piece of DNA and a gene sitting here, with RNA-seq, we can see if the gene is being expressed. With RTAC-seq, we see where the nucleosomes are and where the accessible DNA is. And using some computational methods, we can then sort of predict what sort of transcription factors are involved in keeping this DNA open and what gives this cell its identity. And then I like to think of ChIP-seq and DNA methylation as a way of just decorating this landscape and adding it all together. And finally, by doing Hi-C, we can know exactly which parts of the genome are touching each other. So what this lets us do is it helps us make educated guesses about what's happening in a cell. So rather than saying treatment X induces gene A, we might be able to say that treatment X upregulates gene A through a particular transcription factor and a remodeling of a particular histone mark. And the reason this is nice is that you can then do follow-up experiments. You can look at the specific transcription factor, you can knock it down, you can use antagonists, and you can also use epigenetic mimics to try and block pathways. So in terms of today's talk, with assisted reproduction, but also in general, the epigenome is what carries the memory of the cell. So here on the y-axis, I'm showing the epigenetic signal, and on the x-axis is time. And what's important to remember is that the different molecular marks that are produced in the cell have different half-lives. So let's say we have an exposure A, in this case in vitro culture or infection. We remodeled the RNA, so genes are expressed, active histone marks follow those genes. And after these first signals are gone, it's only then that we start to get the priming histone marks being established, and finally DNA methylation. Once DNA methylation is established, it can last for a long time. So for example, smoking-related DNA methylation can be found 20 years after a person stops smoking. And the reason this memory is important is because maybe are exposed to secondary exposures in the future, we respond differently depending on our memory of our cells. So what this might look like is you might have a progenitor cell that gets shot off course with a particular exposure, and that changes its phenotype and how it responds to future insults. So in the lab, we use human models. We use longitudinal cohorts. So I'll give you just an example here of a human model of disease, in this case, sepsis. So why these models are nice in humans is because rather than having a complicated disease, here you can have longitudinal samples on the same person before and after an insult happened. So for example, here is just an example of 3 donors who went through this infusion of LPS. And you can see that even before they're injected, some people are quite uncomfortable and they become very uncomfortable and feel extraordinary pain during this time period in the sepsis-like phenotype, whereas some people are quite unbothered. If you take an innate immune cell from this individual and we put it in culture and give it a secondary stimulation, these cells don't respond, they're tolerized. This is one of the main killers of sepsis patients after they survive, is that they can't respond to other infections. However, if we give this monocyte yeast sugar, we expose it to this beta-glucan, we can rescue this function. This is what the actual data looks like. This is the naive monocyte. This is after it's been tolerized, and then this is after the function's been recovered. And what's important from my perspective, when I think about this, is that this rescue of function is accompanied by rescue of epigenetic marks. So here is a naive macrophage. Here it is tolerized. And here it is when we rescue it. So we're basically recapitulating the histone marks, in this case H3K27 acetylation, which is an active histone mark. So by remodeling the epigenome and of course metabolome and other things, we regain function in these cells. So how can we do this sort of approach on a larger scale? Well, the answer at our institute at the Royal Children's Hospital is to use clinical trials and cohorts that have longitudinal sampling. And that's the backbone of all of our research. So in a way, these are natural experiments, similar to like infection models, where we have comprehensive environmental and clinical outcome data, and we can study all sorts of things from vaccines to infections to stress, alcohol, etc. And these are necessary to understand how the epigenetic underlies these complex diseases. So here are just some examples of the cohorts we have available. And this is not all of them, there's many, many more. But they cover a lot of different complex diseases and environmental exposures, and these are importantly ongoing. So these are longitudinal samples of children over a long period of time. We also have new cohorts underway, for example, to look at hormones and epigenetics, such as gender-affirming hormone therapy and pregnancy, to see what hormones are doing to the epigenome. But today I'll focus on assisted reproduction, which is the one of the oldest cohorts at the hospital. So this is the CHART study. It's a clinical review of health of adults conceived following assisted reproductive technologies. So it was touched on earlier. ART conception is associated with an increased risk of preterm birth, low birth weight, being small for gestational age. However, in this CHART study led by Professor Jane Halliday, together with Monash IVF and Melbourne IVF, They did a study of 193 adults, so they're between 22 and 35 years old. And interestingly, this population of adults, there was no evidence of increased vascular cardiometabolic risk, and there was no evidence of any respiratory problems. So these are healthy adults conceived by ART. And what we were able to do is, all the adults that were recruited in the study, we were able to go back and get their Guthrie cards at birth. So these individuals were conceived somewhere between 1983 and 1996. And what this allowed us to do is to have matched samples of these individuals as babies and then as adults. So this is the study design. We have at birth the Guthrie cards for 58 controls and 149 ART individuals, and then 75 controls and 158 in adulthood. And we looked at DNA methylation using the EPIC array. So the breakdown of our ART group is 105 IVFs, mostly fresh embryo transfers, and 30 frozen. And we also had the gift group, which doesn't include any culturing. So importantly to notice, we don't have any ICSI in this cohort. And when we look at our sort of ART subtypes, we have the hormone stimulation. In the frozen, there was hormone stimulation, but by the time they're put into the mom, it's a normal hormone cycle again. So the idea there is that the hormone effect is not as big. While the IVF both include the in vitro culture, the gift does not. So what we looked in this cohort is DNA methylation. So the reason for this is that if we were going to see memory in an individual that was born by ART, DNA methylation was the best candidate to carry that information. So DNA methylation is a covalent modification of DNA. It's very stable. It's mediated by DNA methyltransferases, and it's an addition of a methyl group to a cytosine in the context of a CpG site. So the distribution of DNA methylation is interesting. It's dispersed at very low density across the genome, but it's found at very high density at CpG islands in gene promoters. And sort of the classical view of DNA methylation, that it silences genes, comes from a lot of cancer studies in the past, where in almost every single cancer, tumor suppressor genes are completely hypermethylated in the promoter region, and that turns them off. So what we used in this study is the EPIC array. So it targets 850,000 CpG sites out of the total 28 million. Why use the array instead of, let's say, sequencing? Well, it doesn't cover all the CpG sites, but the sort of sensitivity is a lot higher than doing whole genome by sulfide sequencing. And it covers most of the regions we think are important in the genome. So from now on, when I talk about a probe, I'm talking about a CpG site in the genome. So this is the study design. And the very first thing we found when we did this comparison was when we looked at differential methylated probes in babies and adults. We found quite a few at birth, about 2,300, but none of them in adulthood. So the effect of ART, or at least the children conceived by ART, at birth you could see a signature. By adulthood, that signature was attenuated. And when we apply this sort of 5% cutoff, which helps us sort of at least have some sort of quantifiable difference between the ART and non-ART group, we had about 137 differentially methylated probes in babies and none in adults. So what happens to these probes over time, the ones we've found in neonates? So this is a scatter plot. It shows on the x-axis the difference between the ART group and the non-ART group in babies, and on the y-axis the same probes in adults, and the ART group minus the non-ART. So these are cut off here, this 5% line. So these dots, these are all different CpG sites in the genome that are differentially methylated in babies. And you can see that in adults, they do not reach that significant level. So they're basically slowly losing that degree of change with very specific exceptions. And we can also plot that as a bar plot here. You can see that a lot of these red dot bars represent the differences between at birth, and the blue shows the differences in adulthood. So the differences are sort of leaving, with notable exceptions at these genes here, NICAP3 and CHRNE. So probes at CHRNE, this was the most significant region we had. And this is what the data looks like. So what I'm trying to show here is a dot plot of DNA methylation on the y-axis with the controls and ARTs. And what's important to notice is that while the difference is about 6.3% as a group, you can't predict who was born by ART by purely looking at this denomethylation pattern. They're different as a group, as a population. And when we look at these same probes in adults, what you'll notice is that the difference may still be there, as you saw from the scatter plot. The main thing that's happening is that it's not as big and the p-values are not as strong. So basically we're losing that effect. So next what we want to do is sometimes you can find DMPs in the middle of nowhere. We have a lot more confidence that something's important if it's occurring in regions, but this means there are multiple CPG sites changing in the same direction at the same time. This is a summary of what that looks like. So here we have the size of the DMR. So this is how big that region is and how many CPG sites are in that region that are being and are differentially methylated. And here's an example of the ChRNA gene. So this is the transcriptional start site. It goes from right to left. And these are the EPIC probes that cover this gene. And I'll maybe walk you through this figure. So on the y-axis, we're showing DNA methylation. The red is the neonatal non-ART group. The orange is neonatal ART. The dark blue is adult non-ART. And the light blue is the adult ART group. So this gene is actually quite dynamic. So for example, if I highlight this region here, there's no difference between non-ARTs and ART group, but there's a difference between neonates and adults. So this is a age-associated methylation pattern. And what's interesting when we look at the region that actually does have an effect of ART, let's say this set of probes here, we can see that as the person ages, methylation level actually goes from about 20% to 50%. But the difference between the groups is maintained. So they're changing over time, but that ART group is still showing a lower methylation even as the changes are occurring. So at the time, back in 2019, we want to replicate this data. So what we did is we found data from an American study that did a similar analysis, that used a slightly different approach, but on methylation data. And we found that the same region is also hypermethylated in their data set, so a completely different set of individuals. But what we've been able to do since then is, since our paper was published, there's been at least 6 or 7 studies that have come out that have replicated this data. And I would just like to highlight these 2 genes that have been replicated the most times, NECAP3 and ChRNA. These are probes in the genome, in the region. And this is the original study that we published, where we found loss of methylation at both genes in the ART group. We've now been able to replicate some of this in another data set from our children's hospital for the NECAP3 gene. And there have been other studies that have replicated either one or the other. But what I'd like to highlight now is just the heterogeneity of all these different studies. So they're all looking at ART, but if you actually look at what they're looking at, they're quite different. So in our case, we had IVF, we had fresh and frozen IVF and GIFT. And I'd like to highlight this study by Harburg that came out this year in Nature Communications, where they had 962 ART and 983 non-ART individuals. So by far the biggest study. And what they had in this study was IVF and ICSI, about equally distributed, and equally distributed fresh and frozen. Now the interesting thing is, if even though this study is huge, if you were to take it at face value, you would have assumed that these differences were due to culturing, right? Because it seems like they're associated with IVF and ICSI. However, in our data set, we had the exact same differences present in the GIFT group, which is suggesting that this has nothing to do with culture. It's something inherent in the either in the infertility or the stress associated with assisted reproduction. And one other thing I'd like to highlight is when we started working with this BIS cohort, when you get the metadata of these groups, you have a column that says ART, and it's either a 0 and a 1. So if you take it at face value, you say, okay, 1 means ART. Sometimes you have to read what exactly that means. For some of these people, they received IVF treatment, of course. Some received hormones. Some received acupuncture, which I think doesn't really fall within an ART procedure. So what we found in our data is that the DNA methylation signature was independent of culture. So here's a sample breakdown. We had 105 IVFs and 35 GIFTs, and then we had 75 fresh embryo and 30 frozen embryo. And when we look at this top probe in the NECAP3 gene, we see that the loss of methylation is occurring in all the groups. Maybe not as much in the GIF group, but it's still there. And at the CHRNE, definitely in all the groups equally the same. So what this is telling us is that this loss of methylation has nothing to do with culture in this context. So the fact that we see it in the IVF culture, the effect that we might be seeing is probably genetics or the superovulation or the stress associated with IVF, etc. And then another thing that we were interested to explore only for historical reasons, because there's always been an interest in imprinted genes and whether they were more sensitive to change in ART. So what we did is we took all the probes and we only extracted those that sit within 50 imprinted regions, which included 706 probes. And then we just looked at the percentage of those that are different and an unadjusted p-value compared to all probes. So if we applied an unadjusted p-value, about 9% of all probes are differentially methylated. However, we can see that imprinted genes, if anything, are underrepresented in this list. So they're not enriched in this group, and none of them were in the adjusted p-value group. And when we look at their differences, none of them reach 5% in either the babies, at the baby stage, or in adults. So the answer is no. We saw no evidence of imprinted genes being enriched, but I guess it should be noted that these are healthy adults, and none of them had imprinting disorders. However, it's always interesting when you go through the literature is that there was a Nature Genetics paper in 2018 that suggests that potentially CHRNE, our top hit, is actually shows parent of origin DNA methylation patterns. So it is a potentially imprinted gene, but that's something that needs to be explored further. It was an interesting observation. So the future plans for this work is to, well, one, replicate what we found, but the second thing is to disentangle this whole idea that most of the hits that we found and other people are finding since our study was published was to identify which regions are actually susceptible to culture, if any. Because so far it seems that there's an ART signature, but that is not influenced by culture itself. So what we developed was a targeted bisulfite sequencing assay. So this is sort of like whole genome bisulfite sequencing, except that first we do a PCR of a specific region. And the reason this is nice is that we can then screen thousands of samples at a time, and that's the idea that we want to do now. So basically we're, at the moment, trying to get as many collaborators as possible to get blood spots or any sort of cord blood samples from babies born around Australia. And if you have any suggestions or ideas, they're always welcome. So what we'd really like to do is to be able to separate all the different groups of IVF or ICSI or etc., and to identify specific regions that might be sensitive to freezing or hormones, etc. So in conclusion, based on the chart cohort, adults conceived by ART are healthy. They don't have any diseases. When we look at these individuals at birth, we find specific regions of the genome that show DNA methylation changes compared to the non-ART conceived individuals. The vast majority of these DNA methylation changes at birth are resolved by adulthood. So that's interesting. So that just suggests that just sort of the life course accumulation of environmental signals eventually dilutes out that effect that we see at birth. The most replicated regions are in the NECAP3 and the CHRNE genes. This has been replicated several times, like I said, but in every single time, the ART groups are quite distinct from each other. So the only common thing is that they are, in fact, probably infertile parents. And so therefore, it's unclear what the DNA methylation reflects because it's present in all the different individuals. So in larger cohorts, we may be able to disassociate these patterns. And I think that's where it needs to go. And the problem is that a lot of studies are underpowered. And what we really need is we need hundreds and thousands of individuals to be part of these studies, where we both have the culture, the effect of freezing, and also uncultured controls. So I'll finish there, and thank you for your time. That's it. Thanks.